Good afternoon. Today's lecture is part one of a two-part lecture series dealing specifically with Mendelian genetics, or what we oftentimes just generally refer to as transmission genetics. The first two lectures were both an intro, a historical review of genetic mechanisms, or thoughts and ideas that contributed to our modern understanding of genetics, and the second lecture was hopefully just a very good review of mitosis and meiosis, mechanisms that are really important to understand as we transition into today's lecture material. So the figure that you see here is one that you saw in the previous lecture, and I just want to recap the idea that the definition of genetics is the scientific study of heredity and variation. Heredity focuses on how traits are transmitted or passed on from generation to generation, and that is going to be part of what we're going to discuss with this Mendelian genetics lecture series. And then also the variation associated with that. So in this figure, again, a figure you've seen previously, you have two individuals who have produced offspring. The outward physical appearance of the two individuals in question have dark hair, dark eyes, and sort of olivey color skin. The offspring that they have produced is sometimes intriguing to certain individuals. For example, the child on the right has dark hair, dark eyes, and olivey skin, and is sort of what you would expect based on the outward physical appearance of the two parents in question. The other child, the child on the left, has very different outward physical features. She has blonde hair, blue eyes, and relatively fair skin. So this child doesn't look like what we would expect based on the physical appearance of the parents in question. What I hope you will get as we continue on through this lecture series is a good understanding of how, again, traits are passed on from generation to generation, and how we can account for variation, such as the variation that you see in this particular example. Before we can continue on talking about genetics, what we need to do is review a few concepts. The first concept is a mechanism of reproduction. That is, some organisms have the ability to reproduce asexually. What that means is that they produce new individuals that are genetically identical to the original parent. There is no mixing of genetic information from two different sources in asexual reproduction. This is common in single-celled organisms. They reproduce by simple cell division, which is a mechanism of asexual reproduction, like you see on the left-hand side with the amoeba. However, there are multicellular organisms, both animal and plant, like you see on this slide, that have the ability to reproduce asexually as well. Sea stars, for example, can break off one of their arms, and one of those arms can develop into a new individual. Plants are very well-known examples of asexual reproduction, where runners or tubers or additional components can be produced to produce new organisms that are distinct from the original, yet genetically identical to the original. However, when we focus on genetics, we focus on a different mechanism or mode of reproduction known as sexual reproduction. In sexual reproduction, simply what we have in biological terms is the mixing of genetic material from two distinct gametes. In the slide, it's listed here as an example of fertilization of an egg by sperm. However, if you have two distinct gametes that come together, this can also result in sexual reproduction. The point behind sexual reproduction is that you have a mixing of genetic material that allows for a new genetically unique individual to be formed from this mechanism or mode of reproduction. And when we focus on genetics, the concept of sexual reproduction is something that needs to be clearly understood before we continue on with genetic principles. So where we start off talking about genetics is looking at the work of Gregor Mendel. He is considered the father of modern-day transmission genetics, that is, how genes or traits get passed on from generation to generation. It turns out at the time of Mendel's work, there were two hypotheses on how traits were transmitted. The first one is referred to as blending inheritance, and blending inheritance proposed that somehow genetic material was mixed, and eventually you got this mixture in the new individuals of the genetic material. However, with blending inheritance, the concept was that traits were blended together and they were not distinct in the offspring. So, for example, if you had a very tall parent and that individual mated with a very short parent, you would expect all of the individuals in the offspring to be sort of medium or intermediate height, as the two extremes in traits were blended together in blending inheritance. However, what Mendel had proposed in his work is something referred to as particulate inheritance. He proposed that parents passed on discrete heritable units that retained their identities in the offspring and were not blended or mixed together. We will see how he came to these conclusions in just a little bit. One of the other things I always like to mention is that Mendel was one of the very first individuals to analyze patterns of inheritance and document those statistically. It turns out that his work was actually uninterpreted or not well interpreted for a number of years, simply because he added the statistical analysis to it. Mendel's organism of choice was the garden pea. He worked on the garden pea because they were very easy to work with, they were easy to grow, there were many distinct or distinguishable varieties that were readily available with a clear history in ancestry. They can be very easily manipulated, meaning the female and the male components can be separated very easily so that anyone interested in doing so could form crosses that they wanted to do because the male and the female components were very easily identified and separated. It turns out that what he was interested in overall was trying to make new flowers or new flower colors. And from his body of work, again, is the foundations of transmission genetics that we know today. So what Mendel worked on, or the terms that Mendel utilized, was what he referred to as a character and a trait. A character, in Mendel's definition, was a heritable feature that can vary among individuals. The trait is the variant or the variation of a given character. So what you see in this table is an example of the characters and the traits associated with those characters. So for each, on the very far left column, you see the character name, seed shape, seed color, pod shape, and so on. And it turns out that there were two very easily identifiable traits associated with character. So if the character he was interested in was seed shape, it turned out that there were two possibilities for seed shape, round versus wrinkled. If he happened to be studying the character seed color, there were two possibilities there as well, yellow versus green. So again, there were characters and there were traits that Mendel was able to very easily describe and work with. Mendel performed a large variety of crosses. One example is what you see here in this slide. The first thing I want to do is refer to some definitions so that we can understand what exactly it was he did. This is referred to as a monohybrid cross, and simply what that means is that in a monohybrid cross, you look at one character and one character only. So in this particular example, he looked at plant height. Didn't care what the flower color was, what the seed shape was, what the seed color was. In a monohybrid cross, he focused only on one character and looked at only that character. So again, in this example, he looked at plant height. Some additional definitions associated with crosses is the parental generation, designated by the letter P, and this just indicates the individuals that are utilized in the initial cross. The offspring from that cross are referred to as the F1. which is short for first filial generation. The word filial comes from the Latin filius, which means son or offspring, and so you're looking at the first offspring in this cross. In most genetic crosses, the geneticists typically continue on by crossing individuals from the F1 to one another and looking at the next generation, which would then be referred to as the F2. So, in this particular example, he worked with what was known as true breeding plants. That means that the plants in the parental cross came from a long line of plants that only ever produced that specific trait. So the character he was focusing on in this example was plant height, and the two traits were tall versus dwarf. So the tall plant in the parental cross came from a long line of plants that only ever produced tall plants. The dwarf plant in the parental cross also came from a very long line of plants that only ever produced dwarf plants. In this cross, he looked at the F1 and found that every single individual in the offspring were tall, and this caused him some concern in that it seemed like the dwarf trait was somehow lost overall. However, he then took individuals from the F1, crossed them to one another, and looked at the offspring, which is known as the F2. In the F2, he saw plenty of plants that were tall, but he saw the reemergence of the dwarf trait, which seemingly was masked in the F1. Specifically, he actually counted up the number of individuals in this cross. There were a total of 1,064 plants in this cross. 787 of them were tall, and 277 were dwarf. What Mendel did was use a simple ratio calculation of the number of tall plants to dwarf plants. So if you take the number of tall plants, 787, and compare that to the number of dwarf plants, 277, you get a ratio of about 3. What that really means is that for every one dwarf plant, there were approximately 3 tall plants. This is a ratio of 3 to 1. Now, Mendel didn't stop there. He didn't stop at just doing the crosses between tall and dwarf plants. He looked at all of those characters that were listed in the table previously, or a few slides prior to this, and he performed the same type of crosses that we just outlined for the tall and the dwarf plants for all of those other characters. What he found every single time that he did those crosses was the same ratio of 3 to 1. More specifically, in the F1 generation, there seemed to be a loss or a masking of one of the traits. Then in the F2 generation, there was a reemergence or reappearance of that trait that was masked in the F1. And in the F2, again, with those simple ratio calculations that we just talked about in the last slide, he came to the same ratio concept every single time he performed these crosses, and it didn't matter which character he happened to be looking at when he performed those crosses. Every single time in the F2, he would end up with the classic 3 to 1 ratio. Now, Mendel didn't stop there. What he needed to do or decided to do was think about this for a long period of time and then come up with some explanations to the results that he saw. One of the first things I want to add is another set of terms and definition that Mendel himself used. He utilized a term or terms known as unit factors. And what he defined unit factors as are basic units of heredity, and these basic units of heredity are what determine various traits. This, of course, is now what we would refer to as our modern-day gene or set of genes. But in Mendel's terminology, he utilized these types of terms: unit factors, basic units of heredity that determined various traits. So again, what I stated was that he needed to explain or come up with some rationale for the results that he did in fact establish. The first one is that these unit factors that we've just defined must occur in pairs in every individual. If in fact they do occur in pairs, there are 3 different possible combinations that could exist. If an individual had 2 copies of unit factors that were instructions to make tall plants, well, it makes sense, then that would be or result in a tall plant. If an individual had 2 copies of the unit factors that were instructions to make dwarf plants, well, then the plants would be, in fact, dwarf. In both of those cases, you had genetic information or unit factors that instructed the development of the plant one way versus the other. If the 2 sets of instructions or 2 sets of unit factors indicated to be tall, the plant would be tall. And if the 2 sets of instructions or the 2 set of unit factors had the dwarf information, then the plant would in fact be dwarf. There is, however, one additional combination that could occur if individuals do in fact have 2 copies of unit factors. And that would be a situation where 1 unit factor is the instructions to make tall plants, and 1 unit factor is the instruction to make dwarf plants. Now in this figure, I have those factors noted with the letter D, and we have uppercase D indicating or the instructions to make tall plants, and the lowercase d to indicate or our instructions to make dwarf plants. We'll get to that nomenclature or terminology in just a little bit, but for now, let's just assume that that's how these are in fact designated. So again, the last combination of 2, where you have 1 unit factor for tall or tallness and 1 unit factor for dwarfness, is an interesting combination, and we'll get to that in the next slide. Mendel's second principle, or second explanation for the results that he did in fact see, is a concept of dominance versus recessiveness. And this ties into the previous slide where we just talked about a situation where if an individual has 2 copies of a unit factor, and in the case that we just talked about, 1 unit factor was instructions to make tall plants and 1 unit factor was instructions to make dwarf plants, what happens in that situation? What he proposed was that when 2 unlike factors are present, 1 factor is going to be dominant, or is what's going to be seen in the outward appearance of the offspring. He introduced then the terms dominant and recessive, where the term dominant is a term that's applied to the trait or the factor that is expressed regardless of the second factor. It is the one that we see in the offspring. The second term then refers to the other unit factor, which would be termed recessive, and this is a term used or applied to a trait or factor that is only expressed when the second factor is the same. So, in the example that we've just been going through, in a situation where an individual has 1 copy of the tall trait and 1 copy of the dwarf trait, in those situations, the dwarf trait basically is masked and the tall trait is dominant. It's what we see in the offspring. Mendel's third principle, or third explanation for the results that he saw, is a concept of segregation. What Mendel proposed was that if every individual has 2 copies of a given unit factor, that during gamete formation, those paired factors are going to segregate from one another. Now, he used the term segregate. For us, we might change that to separate. Basically, what he's saying is that if you have 2 copies of a given unit factor, that when gametes are formed, those 2 copies have to separate from one another, and each one of those copies is going to go into a distinct or individual gamete. Now we'll look at that mechanism with a simple way to follow that mechanism in the next slide. But what I want to focus on with this particular slide is the concept that if in our parental generation we had true true breeding plants for the tall trait crossed with a true breeding plant for the dwarf trait. The tall plant must have had 2 unit factors that were instructions to be tall, indicated by the 2 copies of the capital letter D. And the dwarf plant must have had instructions or 2 unit factors that indicate that the plant should be dwarf. And on this slide, it is noted with 2 copies of the lowercase d. What Mendel proposed with segregation is that those unit factors have to separate when gametes are formed in those parents, and it is an individual unit factor that gets passed on to the next generation. So in the F1, the only genetic information that the tall parent could pass on was the tall trait, indicated by the capital letter D. And the only genetic information that the dwarf plant could pass on with respect to height is to be dwarf, indicated with the lowercase letter d. So in the F1 individuals, you have individuals that have 2 different unit factors, 1 the instructions for being tall, 1 instructions for being dwarf, indicated by the capital letter D and the lowercase letter d, respectively. In the F1, what he saw was that all of these plants were in fact tall. The dwarf trait was masked, like we mentioned previously. And so in this case, or in this example, this is what is meant by the dominant and recessive combination. If you have 2 different unit factors, one is going to be expressed and the other is essentially going to be masked. If you know the genetic makeup of individuals in a cross, you can then utilize that information to make predictions about the potential offspring. This example that we see here is utilizing what's known as a Punnett Square, and a Punnett Square is just a really easy way to do the bookkeeping to make the predictions associated with a genetic cross between individuals where you know the genetic information. So let's take one step back and define a few words before we move on to what's happening in this particular Punnett Square example. We're going to add in the terms genotype and phenotype. The term genotype refers to the genetic composition of the individual. What alleles are specifically present in the individual for a given character? That is the genotype. The phenotype is the physical appearance that we see due to the genotype. So, back to the concept of dominance and recessiveness, if an individual has 2 copies that are different, one will be dominant and one will be recessive, then the phenotype will be the dominant phenotype. In addition, when we talk about genotype, we can be more specific with respect to the genotype. A homozygous genotype is a genotype where the 2 alleles are exactly the same, and a heterozygous genotype is going to be one where the 2 alleles differ from one another. So, going back to this example, if we have a cross between 2 individuals, one individual being tall and the other being dwarf, again, in Mendel's case, these were true breeding plants, meaning they came from a long line of plants that only ever produced that particular trait. So that goes to say that the tall plant was homozygous for the tall trait, and the dwarf plant was homozygous for the dwarf trait. Mendel's prediction or hypothesis, one of his hypotheses, was that during gamete formation, those 2 copies are going to separate from one another. So that's what you see here with gamete formation. What you can do then with a Punnett Square is list all of the possible gametes that would form from one individual along the side and the possible gametes formed by the second individual along the top. Using the Punnett Square, you can then make predictions about fertilization events, where if the first gamete is fertilized by the first gamete in the dwarf plant, this would be the genetic combination that we would expect. And you can fill in this information continually, making predictions about fertilization events which would then result in a specific genotype outcome in the offspring. So in this particular case, what we see is that all of the offspring, 4 out of 4, are predicted to be heterozygous, and all of the offspring, 4 out of 4, are predicted to be tall. So the genotypic outcome is 4 over 4, or 1, that they will all be heterozygous, and the phenotypic outcome, because of the classic dominant recessive relationship, is that all of these individuals will phenotypically be tall. All right, let's continue that rationale that we just utilized in the previous slide and look at what would happen in the F2 generation. In the F2 generation, you are crossing 2 individuals from the F1. So our individuals in the F1 are both heterozygous in genotype and phenotypically tall. If we then continue on to make a prediction about what type of gametes they would produce, each of those individuals would produce 2 sets of gametes, or 2 distinct types of gametes, with respect to plant height. Those gametes that contain the instruction to make tall plants, and those gametes that contain instructions to make dwarf plants. Now what we can do is consider a cross between these 2 individuals. If the gamete that contained the tall instructions from the first plant came together in a fertilization event with the gamete that contained the tall instructions from the second plant, the offspring that would result would be homozygous for the tall trait or homozygous dominant. But just as likely a scenario is that first gamete from the first plant that contains the tall instructions could encounter the second gamete from the second plant that contained the dwarf instructions. In this case, the individual offspring that would result would be heterozygous. However, you still also need to consider that maybe the first gamete from the first parent is not the one that's taking place or utilized in the fertilization event. What if it's the second gamete from the first plant that contains the dwarf instructions? What if that comes together in a fertilization event with the first gamete from the second plant? And in this case, the individual offspring would again be heterozygous. The last possible scenario is that the first plant, it's the second gamete that contains the dwarf instructions, comes together in a fertilization event with the second gamete from the second plant. In this case, then you also have a homozygous plant that would result, but this would be a homozygous dwarf plant. This is exactly what we are doing when we utilize a Punnett square, and this is just another way to take that into consideration. The Punnett square is a lot less messy, a lot more neat, and much more easy to follow than the arrows. But my point here was to make sure that you understand how we are making these predictions based on combination of gametes in fertilization events. Either way you look at it, with a Punnett square or with this a little more complicated arrow-based mechanism, what you see is that there are the combinations 1, 2, and 3 would result in phenotypically tall plants, and the last combination indicated by the number 4 would result in a dwarf plant. This also then shows that 3-to-1 ratio that Mendel had described. Now that we've established some of these basic principles of Mendel's, utilizing the Punnett square and making predictions about offspring, what we can do is take that one step further in another genetic tool. When we talk about genotype and phenotype in a classic dominant recessive relationship like we've just described, there are 2 genotypes that can contribute to the tall phenotype. Both homozygous dominant genotype and heterozygous genotype, again 2 distinct genotypes, can produce the tall phenotype. So how is it that we can distinguish the genotypes from one another genetically? We can do that by performing what is known as a test cross. And a test cross is always going to be a cross between the individual expressing the dominant phenotype to the homozygous recessive individual. So the test cross portion is crossing the individual in question, the one we want to establish the genotype of, to a homozygous recessive individual. What you see are the two examples that would result in a test cross and how we can work our way back to establishing what the genotype is of the individual in question. So again, in this case, we would have an individual who has the dominant phenotype, and what we're trying to establish is the genotype of that individual. In panel A, if that individual with the dominant phenotype was homozygous tall or homozygous dominant, and we crossed that individual to the homozygous dwarf individual, the only genetic contribution that the homozygous tall individual could make is the tall trait, and the only genetic contribution that the homozygous dwarf individual could produce or contribute is the dwarf trait. All of the offsprings would be heterozygous, and they would be all phenotypically tall. However, in panel B, if the individual in question, again, a individual with the dominant phenotype who we are trying to establish the genotype of, is heterozygous, if you go through the crosses that you would expect, in the end, you would expect with half of your offspring with the tall phenotype, and half of the offspring would have the dwarf phenotype. So the outcomes are different depending upon the genotype of the individual in question. So with the test cross, if you're trying to establish the genotype of the individual in question, you would go through the test cross, you would analyze the offspring, and based off of what the offspring were, you could go back and establish what the genotype of the individual in question is or was. Now, as I mentioned on day one, much of genetics deals with problem solving, looking at problems and trying to establish answers to the questions that are being asked. So what I want to do is take a step back and just go through what I typically do when I'm trying to address or answer a typical genetics problem. So you have a sample problem here that states albinism in humans is inherited as a simple recessive trait. Determine the genotypes of the parents and offspring for the following families. When two alternative genotypes are possible, list both. A, 2 non-albino or normal parents have 5 children, 4 normal and 1 albino. And B, a normal male and an albino female have 6 children, all normal. What I always do with a genetics problem, and I think is a really good strategy, is first to establish some components before you even try to answer the question. One of the first clues in trying to move on with this problem is the fact that there is a simple recessive trait involved in the question. This means that we are dealing with the classic dominant recessive relationship that we just established. As we continue on with our lectures, we will eventually start talking about things that don't fit into these nice, neat, classic dominant recessive patterns. And so you're going to have to look at the clues in the question to help work through the problem to get to the answer. So in this case, we're sort of setting the stage. We know that this is a simple recessive trait. Now, my suggestion and my approach to solving problems is to first establish the gene symbols that you're going to utilize for the problem. What you've seen in the examples previously is the use of the letter D, the capital letter D for the dominant tall plant, and the lowercase d for the recessive dwarf plant. The letter itself typically is the first letter of the recessive trait. This is the convention that is utilized in most situations. So in this case, we are going to establish some gene symbols that we will use for the problem. And in this case, because again, it is a simple recessive trait like I highlighted on the previous slide, the two gene symbols that we're going to utilize is the letter A. The capital letter A is for the dominant, normal, or non-albino trait, and the lowercase letter a is for the albino trait. Again, genetic convention is to use the first letter from the recessive trait. So in this case, we will use the letter A. The next thing before trying to move on to answering the question is again setting the stage for yourself. Establish the genotypes that are going to be possible given the gene symbols that you've just utilized. So what are the genotypes that are possible in any type of situation? Well, we can have homozygous dominant, we can have heterozygous, or we can have homozygous recessive. Two uppercase A's, an uppercase A and a lowercase a, and two lowercase a, all respectively. Lastly, in this establishing phase, I would establish the phenotypes associated with the genotypes we just mentioned. This is a classic dominant recessive relationship, so homozygous dominant is going to result in a normal phenotype. Heterozygous individuals or heterozygous genotypes will also result in normal phenotypes, and homozygous recessive individuals will result in the albino phenotype. So now that you've set the stage with your gene symbols, your establishing of genotypes and phenotypes, now you can go on and start to work on trying to address the question. So part A says that there are two non-albino or normal parents that have 5 children, 4 normal and 1 albino. The parents are both phenotypically normal, as stated in part of the problem for part A. So the genotypes for these individuals could either be homozygous dominant or heterozygous. But an albino child can only result from the homozygous recessive genotype, which means one copy of the recessive A had to come from mom and one copy of the recessive A had to come from dad. The genotypes of both parents must be heterozygous. The genotypes of the normal children are going to be either homozygous dominant or heterozygous, and the genotype of the albino child is going to be homozygous recessive. And there's a quick Punnett Square down at the bottom to show you the cross between the two parents. And again, the key here to establishing what genotype the parents have is because they did in fact have one albino child. The one albino child could only result from getting a recessive from mom and a recessive from dad. All right, so now let's move on to part B. Part B states you have a normal male and an albino female have 6 children, all normal. The first thing I would utilize is the fact that the female is phenotypically albino. The only genotype she can have is homozygous recessive. Again, this is a classic simple recessive trait. The only way you will have the trait is if you are homozygous for those given traits. Point 2 here is that the male is phenotypically normal. Given that the male is phenotypically normal, there are 2 genotypes that he could possibly have, homozygous dominant or heterozygous. You might assume, since all of the children that they had are normal, again, you might assume that the male parent in this case is homozygous dominant. And so this is the Punnett Square on the left-hand side that you might work through, assuming that the male was homozygous dominant, and the female was homozygous recessive. In this case, all of the offspring would in fact be heterozygous and all would be normal. However, when we utilize a Punnett square, it really is just a prediction of the type of offspring we would expect. Given that the male is phenotypically normal, there is a possibility that he is heterozygous, and that's the Punnett square on the right. If he were in fact heterozygous, you could list his gametes across the top, the gametes of the female along the side, and then use that to fill in the information in the Punnett square. In this case, we would expect half of the offspring to be genotypically heterozygous and half of the offspring to be homozygous recessive, and phenotypically half of the offspring to be normal and half to be albino. Now, this can in fact happen. It is based off of probability, but most likely the male in question is homozygous dominant. But you cannot exclude this other possibility. And we're going to come back to this probability concept in just a minute. What I want to do at the tail end of this lecture is really just reiterate this concept of probability. When we talk about the Punnett square, again, we are making predictions about potential offspring based on combinations of gametes coming together. Fertilization is in fact a random event, and that's why we have to consider all possible combinations as possible outcomes, because we don't know which combination is going to take place in a fertilization event. When we talk about probability, let's look at the right-hand Punnett square just in a little more detail. So in this particular combination, what we are expecting is that half of the offspring would be expected to be heterozygous and half of the offspring homozygous recessive. In addition, we would expect half of the offspring to have a normal phenotype and half of the offspring to be albino. But again, what we are doing is predicting the outcome of a given cross. Each cross is independent of the other. So the fact that this couple had one child, we have to consider all of the possible combinations. And each child or conception of a child is an independent event. So just because the first child happens to be normal, it doesn't mean that the second child, if they only had two, has to be albino. It's a 50-50 chance that that offspring will be normal versus albino. So again, it's the concept of probability that I want to keep coming back to and what I want you to think about when you're working with these genetic ratios. What we will do in the next lecture is continue on with these concepts of Mendelian patterns of inheritance and added one additional component, making things a little more complicated, in that we will focus on what's known as a dihybrid cross, and we will work our way up from there.